Welcome to this new lecture of soil science and technology, and in this lecture we will be trying to finish this soil testing topic which we have started in our last lecture. So, in the last lecture we have covered soil pH, soil electrical conductivity, and the soil organic carbon. Now, in this lecture let us start with the very important, that is available nitrogen. Now guys, you know that available nitrogen is basically the summation of both ammoniacal as well as nitrate nitrogen, because plant can avail both forms of nitrogen for their growth. Now this measurement of available nitrogen in the laboratory is basically based on the method given by these two scientists called Subbiah and Asija in 1956. And now remember that nitrogen availability to a plant depends on on its mineralization, and therefore available nitrogen is measured by measuring amount of mineral inorganic nitrogen, that is ammonium and nitrate. And the principle is basically a known weight of soil is treated with an excess amount of alkaline potassium permanganate, which extract easily oxidizable fraction of organic matter. And ammonia, as a result, ammonia is evolved, which is basically absorbed in a known volume of a standard acid, and the excess of that acid will be titrated against a standard alkali using methyl lead as an indicator. So this is the simple principle of this method. And so this let us see what are the reactions which are involved in this method. So here are different methods. So the first step, obviously you can see here, in the alkaline medium, which is basically created by adding sodium hydroxide, these alkaline in condition, this potassium permanganate basically produce these manganese dioxide and obviously this nascent oxygen, and this nascent oxygen basically reacts with whatever organic nitrogen fraction present in the soil. So basically you can see this basically this is a formula of amino acids, and from this amino acids they are releasing the ammonia. So that is why this process is called oxidative deamination, because we are using oxygen here. So in the oxidative deamination process, ultimately we are releasing the ammonia, and remember that these ammonia will be the source of both ammoniacal nitrogen as well as nitrate nitrogen for the plant growth. So we have to basically measure these evolved ammonium. Now this ammonia will be further distilled to form ammonium hydroxide, and this ammonium hydroxide will be reacting or will be absorbed by a standard acid, that is H2SO4, to produce ammonium sulfate, and this is called the absorption step. And in the final step, these unused H2SO4 will be titrated against sodium hydroxide, which is another standard acid, to calculate what is the amount of unused H2SO4. So when we calculate the amount of unused H2SO4, obviously we can calculate the amount of used H2SO4, and from that we can calculate what is the amount of ammonia evolved, and from that we can calculate the available nitrogen in the soil, because all these we know by weight. Now this method is also known as Zeldal method, and this total process in some time is we execute through a Zeldal flask called Zeldal flask or Zeldal assembly. You can see here I have given here, obviously this is a round bottom flask, we call it Zeldal flask, and in this Zeldal flask we take first soil sample, and then we mixed with potassium permanganate and sodium hydroxide obviously, and then we heat it, and ultimately ammonia produce, and this ammonia get condensed within the condenser, and ultimately it absorbed within this known volume of a standard acid, in our case it is H2SO4. So after we absorb and after we collect this ammonia which has been evolved from this oxidative deamination, it is ultimately some amount of H2SO4 will remain unused. So, we will calculate this unused by using some titration against a standard alkali of sodium hydroxide. So, this total process is also known as Zeldal process. So, guys, these are the available nitrogen steps. Obviously, these are very important, and please keep this as a ready reference if you want to do any future testing of soil. However, I am not going to detail all these steps because it is not necessary at this point of time, and you do not need to remember all these steps for your exams and assignments also. Okay. So, let us go ahead and see what are the importance. Let us see about the these are the calculations. So, I have already given the calculations. So, you can correlate the previous slide with this slide and see how we calculate this available nitrogen first in ppm, and obviously this one point I would like to mention here. You can see, first of all, we are when we are calculating the percentage, suppose it is y, and from y to ppm we have to multiply it to 10,000, because 10,000 or 100,000 to get Z, and finally available nitrogen in soil is basically 2.24 multiplied by Z. So, this is very important. Available nitrogen in kg per hectare we have to multiply it by 2.24. Now, the interpretation of this available nitrogen is, if we can calculate the available nitrogen in kg per hectare, which is the final value, is less than 272, obviously that will be low. The medium range is 272 to 544, that is medium. If it is more than 544, it is high. So, basically the interpretation will be, when the soil contains medium amount of nitrogen, we may not want to go for further application. In case of high, we should not apply any further application, because that will be toxic in nature, and in case of low condition only, we will be applying the nitrogenous fertilizers. So, this will be the final interpretation. The available phosphorus, obviously phosphorus is one of the major plant nutrient which plants uptake as primary and secondary orthophosphates. So, H2PO4, obviously ions dominate in acidic range, that is 4 to 7.5, while HPO4 ions dominate in alkaline range of pH, that is 8 to 12. So, both these ions are present at around in neutral condition, both these ions are present in equal concentration at just like here mentioned, that pH 7.2, these ions have equal activity, and Bray and Kurtz number 1 method is useful for acidic and neutral soil. So, we will be discussing this only here. However, remember that for alkaline soil, will be basically we use another extractant which is called Olsen extractant, and so basically now you can understand why we measure pH at the first of any soil testing program, because it helps us to take some major decision for selecting the appropriate extracting agents for subsequent nutrients. So, the principle behind this is, that in the soil phosphorus is basically fixed in 3 major forms. In acidic soil, it is mainly fixed in aluminum phosphate and iron phosphate, and in alkaline soil it is mainly fixed in as calcium phosphate. So, in this Bray and Kurtz method, which is useful for acidic soil, basically we shake the soil with an extracting agent which is called Bray and Kurtz number 1 extractant. So, what is the composition? The composition is basically 0.03 normal of ammonium fluoride in 0.0245 normal of HCl at pH 3.5. So, this is called Bray and Kurtz number 1 extractant. And how it helps? Because it contains both ammonium fluoride and HCl, it basically dissolves the fractions of soil P, that is aluminum phosphate and iron phosphate. So, ammonium fluoride is basically responsible for dissolving the aluminum phosphate and iron phosphate, and HCl results in the dissolution of calcium phosphate in addition to this. However, this is very negligible amount in this pH range. So, this is the principle behind this Bray and Kurtz number 1 method. So, the preparation of reagents, there has to be several reagents you have to prepare, like ammonium fluoride first, then HCl 0.5 normal, then you have to create the extracting solution, then you have to create the Dickman's and Bray's reagent, which is basically ammonium molybdenate, and then you have to prepare the stannous chloride, because this is the coloring reagent. And remember that in this method, basically when we extract the phosphate, we add different types of, we add this stannous chloride for development of a color. Generally, in the presence of phosphorus, there will be development of blue color, and the concentration of blue color will increase based on the concentration of the phosphorus which is present. So, the concentration of the phosphorus will be finally measured through spectrophotometry or colorimetry, and we will discuss that later on. So, in the spectrophotometer, we will create first a standard curve using some standard phosphate solution, and once we create the standard curve, from the standard curve we will be calculating the phosphate concentration of an unknown sample. Okay. So, the steps are given, you have known the principle now. So, the steps are given, weight 5 gram of how to extract and how to create the phosphate solution of known concentration for the standard curve, and how to create the standard curve. All these are given, how to measure the available phosphate, all these are given here. So, please keep this as a ready reference for your future use. Now, remember that once we develop this color, we have to use a colorimeter or spectrophotometer to measure the concentration, and within the spectrophotometer or colorimeter we use a monochromatic light to measure the concentration based on Beer's and Lambert's law. And what is Beer's and Lambert's law, we will discuss later on. Okay. These are the calculations, you do not need to remember all these things at this point of time. However, you must remember that whatever phosphate we get here, available P here in the soil in kg per hectare, if you want to convert into P2O5, which is the standard conversion for measurement of different fertilizer requirement, obviously available P2O5 you can see here, you have to multiply with these factors. So, if you want to convert from P to P2O5, obviously you have to use this 2.29 to convert it to P2O5. Okay. What is the interpretation? Obviously, again based on the available P2O5 in kg per hectare. So, obviously, when it is in between 22.5 to 56, it is considered medium, and when it is less than 22.5, it is considered as low, and when it is greater than 56, then it is considered high. So, this is the, based on this we will recommend whether your test soil has low amount or medium amount or high amount of available phosphate. Okay. Now, the final major macronutrient is I would say final primary nutrient, I would say, is potassium, and available potassium is also very important for measurement of the soil fertility status. So, potassium is abundant in nature, however, plant available potassium is always limited. You know that there are 4 major pools of potassium occur in soil, out of which only exchangeable potassium and clay sites and potassium in soil solutions are available to plants. We have already discussed it in our previous lecture. Now, the principle of potassium measurement is the available potassium in the soil is basically we extract using a neutral normal ammonium acetate. extracted, because the ionic diameter of potassium and the ammonium is almost similar. In case of potassium it is 0.233 nanometer, where in case of ammonium it is 0.286 nanometer. So, both of them can easily replace themselves. So, ammonium can easily replace potassium in the exchange site clay and release them into the solution. So, after extraction, potassium, that is solution plus exchangeable potassium, generally we measure through flame photometer. Flame photometer basically we basically use for measuring the potassium, and it works on the emission spectrometry principle, and the sample basically we introduce into the flame at a constant rate, and filter selects which color of the photometer detects and excludes the influence of other ions. And when potassium in the solution is excited, the electrons from outer orbital of potassium goes to the higher energy states for fraction of a second, and when it returns back to a stable state, it releases the energy which is a specific. So, this is the principle. Now, for that again you have to create a measurement of potassium, you have to again create a standard curve of minimum 5 points, and then you have to prepare the reagents. Here all this procedure is given, and finally the available potassium recommendation will be low, medium, high. You can see if it is varying from it should be 136 to 337.5, then we call it medium, and when it is less than 136 we will call it low, when it is greater than 337.5 we will call it high. So, these are the different grades of available potassium in in kg per hectare. Now, before we discuss in details about the about the flame photometry, which is based on atomic emissions spectrometry, let us discuss about colorimetry. Now, colorimetry we basically use for measuring the concentration of phosphate, and colorimetry basically depends on some principle of relationship between percent transmittance and light path length and concentration. So, let us first see what is the basic parts of a colorimeter, then it will be lot clear to us. So, if we go back and see what is the component of a colorimeter. So, in the colorimeter, first of all there will be a light source, and from that light source there will be further a collimator or lens which will basically align all the light source directly into the monochromator, which is prism or grating, and from that monochromator, prism or grating, that will grade the light, and finally there will be a wavelength selector which will select only a particular wavelength. That is why we call it monochromatic radiation. Now, this particular wavelength will go through a particular sample. So, for example, we have extracted the phosphate and we have developed we have extracted the soil phosphorus, and then we have developed the intense blue color by adding the stannous chloride. Now, the concentration, for measuring the concentration of that blue color or the phosphate, you have to basically take that solution into a cuvette, which is basically quartz or fiber made container, transparent container, and then basically what you have to do, you have to basically we are taking the sample solution here. So, the monochromatic radiation is going through the sample solution. So, I0 is basically incidence radiation and IT is basically transmitted radiation, and ultimately finally there will be a detector, and this detector will finally digitally display the result, or basically this photocell will convert this the intensity of the radiation into electrical energy. So that is why that is how a colorimeter works. So that's why we generally add different solutions here, and based on their concentration we will get their different transmittance or absorbance. Okay. Now, what is the basic principle behind this? What is the basic method, basic principle which governs this colorimetry? This is governed by Beer's and Lambert's law. So let us see what is Beer's and Lambert's law. Let us go back. So the colorimetry says this is the relationship between the length of the solution or path length and the percentage transmittance. So obviously as the length increases, the percentage transmittance decreases exponentially. Similarly, if the concentration increases, then the percentage transmittance decreases exponentially. So transmittance, it is IT by I0, which is transmitted radiation by intensity of the incidence radiation, which is generally we can denote by this, where alpha is an extinction constant and C is the concentration and I is the light path length. L is the light path length. Okay. So you can now one thing is very clear, that depending on the path length, that means the width of that cuvette and concentration of the chemical which is present within the chemical within that cuvette, this transmittance will vary. Okay. So what we are doing? So the absorbance is basically increases linearly with concentration. So as the concentration increases, obviously it will absorb more light and it will transmit less amount of light. So here it is called the Beer's. Okay. So when it is depends on the length, obviously. So when it depends on the length only, then we call it Lambert's law. And when it depends on the concentration, we call it Beer's law. Now, in case of cuvette, you see we are only changing the concentration of the liquid inside the cuvette, because the path length is always fixed, because the cuvette size is almost fixed. So if we combine both Beer's and Lambert's law, then we are only varying the concentration by keeping the length of the Lambert's keeping the length of the path constant. So that's why we call it Beer's and Lambert's law. So you can see here we are taking the log of I0 by IT, and then you can see it is alpha CL, and then it is log 10, converting it to log 10 we are getting eta and then CL. So here basically the Beer's Lambert's law says that A equal to absorbance will be equal to the absorbance will be proportionate to the concentration and length. So this is a combination. So in the Beer's and Lambert's law, obviously when we are applying this Beer's and Lambert's law to colorimetry or colorimeter, we are nullifying this L and we are only playing with this C, that is the concentration inside the cuvette. So that's why we measure the concentration, that's how we measure the concentration of the liquid inside the cuvette, and that's why we measure the concentration of phosphate. So basically we take some standard solution, and using the standard solution we make a standard curve, because we know their so for example if you take 0 ppm and then for example 1 ppm, 5 ppm, 10 ppm up to 100, and then we get their absorbance. Obviously as we increase the concentration, their absorbance will increase. So we will draw a standard curve, and from the standard curve if there is an X sample for which we do not know. So, we will extract that using the standard process, and then we will use the standard curve to get the concentration from here, because we will be getting the absorbance directly from the colorimeter, and by plotting that absorbance value we can directly measure the concentration. So, this is the basic principle behind behind this colorimetry. So, another important thing is flame photometer. Now, using the flame photometer, basically we measure sodium, potassium, lithium and calcium, and basically this is a flame photometer. It is a controlled flame test with intensity of the flame color quantified by photoelectric circuitry. Remember that it works on atomic emission spectrometry and the samples or spectroscopy, and the sample is introduced to the flame at a constant rate, and the filter select which colors of the photometer. So, the basic principle is, first of all, we take the extracted solution which contains the potassium ions in the extracted solution. So, there is an aspirator here, you can see this is an aspirator. So, this aspirator will suck this solution, and inside a chamber it will create an aerosol, and for creating the aerosol you need some kind of air. So, air is inserted from outside through this air pump, and from this air pump it will create an aerosol, and this aerosol will be further vaporized in in contact with a flame, and there will be a flame inside, and the flame will be, and the fuel for this flame is basically LPG or liquid petroleum gas. So, using the flame energy, these aerosol will, the potassium which is present in this aerosol will be going from will be showing some thermal dissociation in flames. So, it will be if there is an any electrons which is present in the ground states, it will be go to the exciting states, and then from the excited states when it will revert back to again its ground states, there will be some amount of energy in the form of light, and the particular wavelength of this emitted energy is basically very specific for a specific element. So, by measuring that particular concentration of that or the intensity of that emitted light, we can measure what is the concentration of that particular element within the solution. So, this is how a flame photometer basically works. So, for individual element, the emitted wavelength range is different. You can see in case of potassium it is 766, in case of lithium it is 670, calcium 622, sodium 589, barium 554, and obviously observed color of the flame will also vary depending on the different elements. So, that is how it measure different these important elements using this flame photometry method. So, if you see the inside curve, the schematic of the inside view of a flame photometer, obviously you can, there is an nebulizer or aspirator which will aspirate, and further it will mix, and there is a mixing chamber, and in the mixing chamber it will create an aerosol and mix, and further this will go to the burner. So, this is a burner and there is a gas inlet, so there will be a flame. So, ultimately it will go to the flame and take the energy, and finally the emitted radiation will be going through this lens and this filter, and finally photodetector, and this photodetector will ultimately convert this this light signal into electrical signal through amplifier and readout. So, this and waste material will be going through this waste U tube. So, this is the inside view of a flame photometer. So I hope guys you have learned something new in this lecture, and we'll be stopping here, and in the next lecture will be still couple of slides are left for soil testing. So I'll be trying to finish wrap up those slides in the next lecture first, and then we'll go to a new topic. Thank you very much.